To follow cholesterol in human body, first we have to go through the fat transportation, which happens mainly in blood, but we need to acknowledge that also some of the fat that we receive through our diet will be first absorbed into the lymph. In blood and lymph, we do have water, and we know large lipid molecules such as triglyceride, which is the main lipid in our diet, are hydrophobic. These are non-polar molecules. They are not water soluble. So the only way that we can transport these large hydrophobic lipid molecules in watery environment of blood or lymph would be some cells must place these hydrophobic lipids in an envelope that is made mainly of protein and some phospholipid and cholesterol. So if we picture how large lipid molecules are transported in blood, as you can see in the image, those cells that I'm going to explain which cells they are, place these molecules at the center of a spherical particle, and the shell of this particle is made of proteins that their job mainly is to make this particle water soluble, and we name these proteins apoproteins. And also in this shell, we can find some phospholipid and cholesterol. And we name the particle which is responsible for delivering these large hydrophobic lipid molecules lipoprotein. The reason is that it's made of protein and different types of lipid. We have 4 major lipoprotein in our body. The largest one is chylomicron, which forms in the cells that line our small intestine. And as you can see, in chylomicron, we have the highest proportion of triglyceride to other 3 components. The heaviest of these lipoproteins would be high density lipoprotein, simply because this is the one that has the highest proportion of protein to the other 3 components. And HDL, high density lipoprotein, is the one that is referred to as good cholesterol, and I'm going to explain why this one is considered good cholesterol, but low density lipoprotein, LDL, is considered bad cholesterol. And let's keep in mind that we also have a lipoprotein which is called very low density lipoprotein, and as you see, in all 4 lipoproteins we find cholesterol, but the proportion of cholesterol to the other component would be different. The highest proportion of cholesterol to the other 3 components is found in LDL. So let's start talking about where exactly each one of these lipoprotein forms and what the goal is. Then it makes sense what happens to the cholesterol that we are following in this video. First, we go to the master of absorption of nutrients, which would be small intestine. In the lining of small intestine, we have epithelial cells that one of their expertise would be absorbing nutrients. That's why we name them absorptive cells. And when we focus on one of these absorptive cells, we see that when the cell absorbs large lipid molecules, eventually it builds triglyceride, because triglyceride is hydrophobic. The cell places triglyceride at the center of a lipoprotein. The shell of this lipoprotein, that is called chylomicron, forms by phospholipid, protein, and cholesterol. So some of the cholesterol molecules that we absorb through our diet are found in the chylomicron. And we know chylomicron is very large. It cannot pass through the wall of the blood capillaries that we have in the lining of small intestine. That's one of the main reasons that chylomicron first will be absorbed into the lymphatic capillaries that we have in the intestinal villi, and we know that we name these lymphatic capillaries lacteals. So chylomicrons are released into lymph that we have inside the lacteals, and we know from small intestine through intestinal trunk, the chylomicrons are released into the left lymphatic duct, or we can name it thoracic duct. When I follow the chylomicrons that travel in thoracic duct, eventually we get here near the junction of left subclavian vein and left internal jugular vein. Those chylomicrons are released into bloodstream. And please keep in mind that when chylomicrons are released into blood, blood has a milky appearance due to the presence of the chylomicrons, and also blood becomes slightly more viscous. So now chylomicrons are found in bloodstream. We follow the chylomicrons in bloodstream, and we see in some places such as liver and adipose tissue, fat tissue, most of the chylomicrons chylomicrons are removed from bloodstream. The reason is when we look at the blood capillaries that we have in liver or adipose tissue, we notice that in the endothelium of these blood capillaries, recall endothelium refer to the simple squamous epithelium that lines the blood capillaries. In the endothelium of these blood capillaries, we see an enzyme that is called lipoprotein lipase. When chylomicrons get here, these lipoprotein lipase enzymes start breaking down the triglyceride molecules that we find in chylomicrons. The result would be fatty acids and glycerol that will be sent into the adipocytes or the liver cells, depending on where we are. Usually about 10 minutes after absorption of chylomicrons, about half of them are removed from bloodstream by liver and adipose tissue. And in a healthy individual, 2 to 3 hours after absorption of chylomicrons, most of them have been removed from bloodstream. We end up with very small amount of chylomicrons left in bloodstream, and obviously when most of the component of chylomicrons go into these cells, what is left would be some smaller particles, and these particles through bloodstream they go back into the liver. So now liver has some of those, for example, cholesterols that we absorb through our diet, and also we know liver cells are very creative. They can produce many different products, including new cholesterol molecules and triglyceride molecules and some other type of lipids. So now liver cells, they do some sort of packaging, place these lipids in a particle that is called very low density lipoprotein. Very low density lipoprotein contains those lipids that liver cells produce, including the triglyceride molecules, and as you can see, we do have also some cholesterol here, and they transport these lipids to mainly adipose tissue. The goal is to send especially the triglyceride molecules to adipose tissue so adipocytes can store fat. When we get to the adipose tissue, clearly we know in the endothelium of these blood capillaries we do have lipoprotein lipase, and as I explained, they're in charge of breaking down the triglyceride molecules and removing these molecules from bloodstream, and the result would be some smaller particles that mostly go back to the liver. Why we send them back to the liver? Because in general, when we want to excrete some lipid molecules that are extra, liver cells are supposed to do the final packaging and prepare them for elimination from the body. However, about 1/3 of these particles transform into low density lipoprotein. In low density lipoproteins, we have a very high proportion of cholesterol to the other three components: triglyceride, protein, and phospholipid. In general, LDLs carry about 75% of our total cholesterol in blood, and this is the primary delivery system of cholesterol to the cells. Why cells need cholesterol? Many cells can use cholesterol to repair their membrane. Also, some cells that we have in, for example, testes, ovaries, adrenal glands, use cholesterol to build steroid hormones. However, it is important to note, to remove LDL from bloodstream, we need to have some proteins in the membrane of cells named LDL receptor. So LDLs should bind to the LDL receptors, and then through receptor-mediated endocytosis, LDLs get inside the cell, and then cell has the enzymes that break down LDL to smaller components. When cells have enough cholesterol for whatever reason that they need cholesterol for, which I explained a few of those reasons, a negative feedback system starts, which results in refusing to build any more LDL receptor. So it's very simple. Cells have enough cholesterol, they do not build LDL receptor, and without the presence of the LDL receptors, obviously the remaining LDLs cannot get into our tissue cells. They are left in bloodstream. That's why we explain that how much LDL is removed from blood depends on the number of LDL receptors that we have in the membrane of tissue cells. How do we end up with a high amount of LDL in our bloodstream? One simple explanation is if our diet contains too much cholesterol, the daily recommended intake for cholesterol is as minimal as possible, simply because some of our cells, including liver cells, can synthesize cholesterol. So cholesterol is not really an essential nutrient for us. It means that we don't need to have cholesterol in everyday diet. The other reason would be in some individuals, genetically speaking, they lack that ability to produce enough LDL receptors. So we have a variety of reasons that we end up with high LDL level in bloodstream. And as I explained in the video, atherosclerosis and LDL cholesterol that would lead to formation of those plaques in the wall of our blood vessels. In general, in healthy adults, LDL cholesterol level that is below 130 milligram per deciliter of blood is desirable. Now, it hopefully makes sense why in those individuals that they have high cholesterol level, we prescribe different drugs, that some of those drugs promote excretion of bile. Why? Simply because the extra cholesterol that is left in tissues or in blood must be sent back into the liver. What liver cells do, they add these extra cholesterols to bile. They use them, for example, for formation of some of the bile salts. Bile will be released into the small intestine, and we can excrete these extra cholesterol molecules as one of the component of feces through the digestive canal. So when a drug promotes excretion of bile, obviously that would be an attempt to help eliminating extra cholesterol from the body. Some other drugs are going to block formation of those enzymes in liver cells that are responsible for formation of cholesterol. So these are some options that we have. In short, we have to collect the extra cholesterol from different parts of our body and send it back to the liver. And liver actually tries to help this process. It produces a lipoprotein that is called high density lipoprotein. The protein proportion in HDL is the highest among the 4 lipoproteins. It is interesting that when liver cells produce HDL, we see that this particle does not contain lipid at its core. Think about the HDL that liver cells produce as a ball that the central portion of this ball is empty. When HDL travels in bloodstream, it collects cholesterol from tissues, from blood, and also from those atherosclerosis plaques that already been formed. And when it's filled with cholesterol, then it's time for HDL through bloodstream to go back into the liver. Liver is going to use this extra cholesterol that it receives in the formation of bile, and that allows the digestive system to eliminate and excrete the extra cholesterol out of body. Now hopefully it makes sense why we consider HDL good cholesterol when the amount of HDL is at a desirable level. That simply means liver is doing a great job in producing enough HDL in order to collect the extra cholesterol from different sections of our body. In a healthy adult, the amount of HDL cholesterol that we are hoping for would be above 40 milligram per deciliter. And now we can talk about the ratio that we use to predict the risk of developing coronary artery diseases. As I explained in the video, atherosclerosis and LDL cholesterol, if plaque forms and blocks any of the branches of the coronary arteries, the consequence would be heart attack, the death of the cardiac muscles that are not receiving enough oxygenated blood due to the blockage of the coronary artery because of the presence of the plaque. The ratio that we use would be total cholesterol to HDL cholesterol. We already learned about what is the desirable amount for HDL cholesterol. In healthy adult, the total cholesterol in blood should be under 200 milligram per deciliter. Let's say in an individual, the total cholesterol is 150 milligram per deciliter, and the amount of HDL cholesterol is 50 milligram per deciliter. So the ratio that we get is 3. Any ratio more than 4 would be considered undesirable. The higher this number is, the more risk the individual has for developing the coronary artery diseases. I hope you find this helpful.